Hello students, welcome to EPG Patashala. I am Dr. Gaurinath from School of Life Sciences, Jawaharlal Nehru University. Today we are going to talk about on module on fibrous proteins. Here I will describe about the structures of alpha-keratin, silk fibrin, collagen, uh, and this chapter is from the biomolecules and their interactions. Here we will learn about the structures and assembly of fibrous elongated proteins, specifically the structure, function, assembly of alpha-keratin, collagen, and silk fibrons. The fibrous proteins have elongated shape. They serve as a structural material and provide structural support to the cells and tissues. By cross-linking and interleaving, different properties can be obtained, and these are served as different functions. Fibrous proteins are usually divided into three different groups. The coil containing proteins of alpha helices like keratin and myosin, the triple helix like collagen, the beta sheet containing amyloid fibers like silks. The alpha helical fibers of wool are flexible, elastic, and can be extended twice of their length, whereas collagen fibers are strong and relatively rigid. The beta sheet fibers are strong as well as flexible. Example, spider web. Some of the spider web are actually stronger than steel of the same dimension. If you take the thickness of the spider web of the same steel, actually the spider web is more stronger. At the same time, spider web is flexible, whereas steel is not. The fibrous beta sheet structures are also formed by misfolding of soluble globular proteins, and these causes diseases like Alzheimer's and prion diseases. Alpha-keratin is one of the classical example of alpha helical rich fibrous protein. Keratin protects epithelial cells from damage or stress, the key structural material making up the outer layer of human skin. And this is a structural component of hair, nails, and which are coded by almost 60 genes. The sequences reveal that there are two distinct but homologous keratin families, named as type 1 and type 2. Keratins contain a central region of 310 amino acids and 4 segments of alpha helical regions, which are separated by 3 short linker segments, predicted to be a beta-10 conformation. The 4 helical segments are made up of heptad repeats like coil-coil regions. These coil-coil regions are, which we have discussed in supersecondary structures module, where the 7 amino acids are repeated. In that, every first or fourth amino acid could be hydrophobic. I will discuss again in the next slides. Here I show you the structural domain assembly of keratin. What I said before, the long keratin molecule, where we have shown the green color segments, which are helical regions, and those gaps which could form turns. And N and C terminal form small globular domains. These ones form the helical regions, form coil-coil dimers, and these dimers come together to form protofilaments, and these protofilaments assemble together to form a bundle of 8 protofilaments to form long intermediate filaments. Alpha-keratins are rich in cysteine. The covalent disulfide linkages between adjacent polypeptide chains, leading to fibers that are insoluble and resistant to stretching. So the more disulfidages, the keratins are more stronger. The central regions form coils leading to a rod-shaped structure with 2 globular domains at each end. These rod-shaped structures interact with each other at the center to form uniform core. These rod-shaped structure, which consist of heptad repeats, where 1 and 4th residue generally have hydrophobic residues, and uh, 5th and 7th residue generally are charged residues. The globular domains of different sizes protect from the surface. They are at the two terminals. The hair is composed of dead cells, each cell packed with keratin microfibrils. Alpha-keratins are rich in cysteine. The covalent disulfide linkages between adjacent polypeptide chains leading to fibers that are insoluble and resistant to stretching. So the more disulfide bridges, the keratins are more stronger. The central regions form coils leading to a rod-shaped structure with two globular domains at the each end. These rod-shaped structures interact with each other at the center to form uniform core. These rod-shaped structures, which consist of heptad repeats, where 1 and 4th residue generally have hydrophobic residues, and 5th and 7th residue generally are charged residues. The globular domains of different sizes protect from the surface. They are at the two terminals. The hair is composed of dead cells, each cell packed with keratin microfibrils. Disorders of keratin lead to several diseases because these are the main building blocks of hair and nails. Thus, its disorders lead to significant deterioration in the mechanical integrity of these tissues. Like there are several disorders one can look in the web, but I have listed a couple of examples. Mononelethrics is a rare autosomal dominant hair disease that results in short, fragile, broken hair. The other disease, epidermolytic hyperkeratosis, which is a rare skin disease, affects about 1 in 2.5 million people, and this is causes due to mutation in keratin 1. Most of these diseases are very rare diseases, but these are genetic disorders which causes, when it occurs, there could be severe problems. Collagen. It is most abundant protein in human body. In our body, it consists of about 25 to 30% of our body weight. It is generally forms bundles as a tight parallel fibers provide strength to tendons and a skin. It is also found as a gel that serves to strengthen the structures as an extracellular matrix. For example, vitreous humor of the eye. The collagen fibers in bones are arranged at an angle to each other to give strength from all directions. Collagen constitutes 1 to 2% of the muscle tissue and accounts for 6% of the weight of strong or tender muscles. The fibroblast is the most common cell that creates collagen. The tensile strength of collagen results due to triple helix structure of the collagen and the assembly of tropocollagen subunits into a fiber. There are several cross links in this fiber that actually makes it more tensile strength. Collagen is formed from tropocollagen subunits. The triple helix in tropocollagen is highly extended and strong. In collagen, nearly one-third of amino acids are glycines. Almost every one-third of the residue is glycine. And proline and hydroxyproline together form another one-third. And rest of the one-third residues could be other residues. But many prolines are actually modified. So the modified prolines are like 4-hydroxyproline, 3-hydroxyproline, or 5-hydroxyprolines. And these proline and hydroxyproline together nearly makes one-third of amino acids. In the right side, I have shown you the picture of collagen assembly. You can see that glycine, X, Y, these are the repeats, the tri-amino acid repeat, which generally see in the collagen central part, where first amino acid is one-third amino acid is always glycine, and X and Y could be proline and hydroxyproline, and those are another one-third of it, and the other one-third could be any other amino acids. These three polypeptides come together to form triple helix formation. And once this pro-collagen is formed, the ends are cleaved by different enzymes, and the central part, the collagen, becomes stable, independent triple helix structure. Gelatin is used in food and food industry is a collagen that has been irreversibly hydrolyzed. So it basically becomes a protein-rich source. Collagen also has many medical uses in treating complications of bones and skin. These are synthesized as long precursors, about 1000 amino acids, called procollagens, with global extensions about 200 amino acids at the both ends. The procollagens are transported into endoplasmic reticulum, where they are hydroxylated and then assembled into triple helix. So three polypeptide strands each has the conformation of left-handed helix, and do not confuse with alpha helix as right-handed helix, to form a collagen fiber. These left-handed helices are twisted together into right-handed triple helical conformation, or you call as a superhelix, a cooperative quaternary structure stabilized by many hydrogen bonds. So there is a hydrogen bond between the different polypeptides. Whereas in alpha helix, you see the hydrogen bond within the polypeptide. In the collagen, you see hydrogen bonds between the polypeptide. Each collagen fiber has several micrometers long and 50 to 200 nanometers in diameter. So these filaments assemble together to form bundles. You can see the right side of the image where the collagen bundles are seen, and these are extrapolated from the electron micrograph pictures of collagen fibers. Collagen polypeptides have large number of repeat sequence. As I described before, the pattern of amino acids are glycine, X, Y. So glycine, the 1/3 of total amino acids in collagen is glycine, and X and Y residues are generally proline and hydroxyproline. So these together are another 33% or 1/3 residues. So these X and Y are not always proline and hydroxyproline. So they are another 1/3 times it's different amino acid. So the extended left-hand helix is about 3.3 angstrom residues per turn, and the raise per each turn is 9.6 angstroms, and per each amino acid is 2.9 angstroms. So it is very extended helical structure. Of course, this is not as extended as beta sheets, but it is not as coiled as alpha helix. It is quite extended helical structure, and this triple helix repeat is about 100 angstroms. So the triple helix repetition you only see after 100 angstroms length. That is the axis of triple helix. Before I talk about this one, I would like to talk about the how a collagen model was proposed. You may know that Professor Ramachandran was the first one to propose the collagen model, where the 3 residues per turn is proposed. The left side one is the initial collagen model proposed by Ramachandran. So every third residue which comes in the center, and that is glycine, and other XY residue could be the other one. So according to him, these 3 coils come together, and then in the center the glycine comes, the other 2 residues on the outskirts. So when Ramachandran was working on collagen model, so initially people say that Professor J.D. Bernal was visiting India that time, and he suggested Ramachandran that why don't you work on collagen, because by that time Linus Pauling already proposed the model of alpha helix, beta sheet, and Watson Crick already proposed the model of DNA structure. So then the molecular composition was exactly not known, except that 1/3 amino acids are glycine, and another 1/3 amino acids are proline and its derivatives, and there are not many technologies available as we have today. So on the basis of diffraction data, Ramachandran initially proposed the what we have, which I'm showing you, the left side. And later, several people, including Crick, proposed the model, And now the final accepted model is the right side, where it's actually the 3.3 residues per ton. So every third amino acid doesn't come exactly at the center, because we have a supercoiled structure of repetition of 100 angstroms. So there is a 3.3 residues per ton. So the next glycine on the next helix comes after a few degrees, which is listed here, and the residues per turn becomes 3.3 residues per turn. So here, where the difference between the Ramachandran model and other models which came later. So people say in India, if you ask anyone who first gave the collagen model, they always say Professor Ramachandran. But in the West, when you ask people who gave the collagen model, they always say Crick gave the first model. So what people say in India, and what the historical fact seems to be, is that when Ramachandran proposed this model and the paper was sent to revision, and it was sent back saying that if all the three residues come in the center, there will be enough crowding and there will be steric clashes where they cannot accommodate. When these comments came, and that's when Professor Ramachandran worked on Ramachandran plot and showed that there is enough space for glycine to accommodate, and glycine can have many conformation possible. And that's how he validated his collagen model. So this is a triple helix conformation, which is now what it's reported. So you can see those three polypeptides in three different colors, where generally the hydroxyproline and proline, which is coming out of the on the surface of the helix. In the center, the glycine molecules are there. So the triple helical structure for collagen with one interchain hydrogen bond per tripeptide. So for every three amino acids, there is one interchain hydrogen bond. So there are three chains, so it will have a three hydrogen bond between three chains. The sequence is glycine, proline, hydroxyproline, which this crystal structure has been determined, and where it is showing three different colors. Triple helix hydrogen bond formed between separate chains. A helix hydrogen bond formed within the same chain. So as I said before, in the alpha helix, what we see is hydrogen bond is within the same chain, where every 3.6 residues, every 1 to 4th residue, you have a hydrogen bond and 3.6 residues per turn. In the case of collagen, it is a hydrogen bond between the chain 1 to 2, 2 to 3, 3 to 4, and that 4 is again a first chain. So in the triple helix, every third amino acid is close to the center of the axis, where there is no space and only glycine can accommodate there. Any other residue, if it comes in the center, it deforms the triple helix. The hydrogen bonds are formed between the NH group of glycine and carboxy group of proline of the next chain. Here you can see the collagen fiber bundle is formed by assembly of tropocollagen molecules aligned side by side. So there's several of collagen fibers together. And if you take one fiber, you can see in the colored where your triple helix is there. And you can zoom in that, and you can see the three polypeptides, yellow, red, and green. Those are coming. And if you zoom in further, you can see the glycine, proline, and hydroxyproline in the center. All the glycines are coming, and hydroxyproline is generally on the surface. Here, the actual crystal structure of collagen polypeptide, where three polypeptides are crystallized as a collagen fiber, and in the right side, it is shown small section of collagen where hydroxyproline to glycine 12 in A chain, and corresponding to proline 37 to threonine 41 in B chain, and glycine 66 to isoleucine 70 in C chain are shown. So other than what earlier predicted, the hydrogen bond between the polypeptides, what is seen in the crystal structure is also water is interacting, and water is interacting with the side chain from one to the main chain of the other chain. So this seems to be is actually responsible for the extra strength of collagen fiber, where one extra hydrogen bond is seen between the polypeptides through water. Here the Crystal structure was determined with three polypeptides. One is glycine, proline, proline. Another one is glycine, proline, hydroxyproline. So both the structures are exactly identical. There is no difference between these two structures. So the hydroxypro or pro containing triple helix collagen structures look exactly same, and they prefer this kind of formation. Water-mediated hydrogen bonds between inter and intra-chain triple helices will differ, thus stability of triple helices must be different. There are different post-translation modification and cross-links seen in collagen molecules. As I said before, the collagen molecules are coated by several genes, and here these collagens are classified into according to these groups. I have listed few of them here. The type 1, which is responsible for the tissues of bone, skin, and tendons, and type 2 is cartilage and vitreous. Type 3 is made up of blood vessels, scar tissue, uterus wall, etc. And type 4 is basement membrane lens capsule. Type 5 is cell surface and exoskeleton. Type 6 is aortic intima, placenta, kidney kind of tissues are made up of type 6 collagen. So few of them I am listed here. You can look into the literature, and they also have a special characteristics. Each one has a special characteristic. They either they are carbohydrate linking, or their solubility, or molecular weight, the composition of other amino acids like hydroxylysine changes from one type of collagen to the other type of collagen. Disorders of collagen. So as we discussed, the collagen is a building block for several types of skin and other tissues. So the disorders lead to significant deterioration in the mechanical integrity of these tissues. So there you can actually, in the literature, you can see many disorders related to mutation in collagen. So here tell you one simple example is Ehlers-Danlos syndrome. My pronunciation may be wrong. This syndrome, which explains the muscle-skin junction disorders. The osteogenesis imperfecta is another disorder which is responsible for brittle bone disease, and this can also cause short height, hearing loss, etc. So these mutations in some of these collagen molecules can lead to several diseases. Again, because these are genetic diseases, which are generally non-curable, and these occur very rare diseases. These are one of the few very rare diseases which can happen 1 in a million, 1 in 10,000 kinds of cases. Now we will discuss about the beta sheet containing fibrous proteins, the silk fibrin. The spiders generate 7 distinct fibers by drawing liquid crystalline proteins from a set of separate gland spinner net complexes. The proteins produced inside these glands are called silk fibrins. The proteins are sorted in highly concentrated, almost 50% of the concentration solution in these glands, mostly in alpha helical conformation. This solution is passed through spinning machinery and mixed with other components, producing a variety of different fibers and beta sheet structures. Spiders can produce different types of silk with different strength. Example, dragline fiber, a spider uses to hide, has remarkable mechanical properties. Dragline fiber is stronger than steel but can bend, contract, and stretch. Silk fibrin genes code for a similar sequence pattern comprising of N and C terminal variable domains. Most of the central part, large repetition of 8 residues of polyala and gly-X regions, where X could be serine, threonine, or glutamine. The bulky regions with valine and tyrosine interrupt the beta sheet and allow the stretchiness. This middle region varies in length in different genes, and the length of these genes could be up to 800 amino acids. Spider silk fibrin arrangement is seen in this figure. So you can see here the spider web, which is in the part A, small one. If you zoom in, it is made up of several silk fibrils, and these fibrils are, if you further zoom in, has a jelly, a mesh kind of structures in which there are crystalline particles, and these crystalline particles are made up of beta sheet nanocrystals, and these beta sheet nanocrystals are polypeptide containing a lot of beta strands. So some parts of the microcrystal structures, other regions form a less ordered mesh-like structures where this crystal part is embedded. The crystal part which gives the strength to these fibers, and this mesh-like structure gives the flexibility. Both together it gives very dynamic structure, both flexibility and the strength. The structure of this silk fibrin beta sheets is, if you see here, three-dimensional view of the stacked beta sheets. The intercalation of side chains like alanine, serine from one polypeptide is intercalated into the other side of the beta sheets. So the beta sheets are stacking in two ways. One is the hydrogen bond between the two beta sheets. So the side chains, which are the perpendicular, those are stacked with the other beta sheets. So this is a stacking of the beta sheets, both hydrogen bond and intercalation of the side chains with the neighboring beta sheets. So it almost forms a three-dimensional structure of beta sheets. Fiber diffraction studies indicated that some proteins in spider milk fibers adopt beta sheet structures very similar to common silk. The fibers contain small microcrystals of ordered regions inserted in the matrix of less ordered regions, this jelly type structure. And this is seen in both EM studies and NMR studies. And each microcrystals are about 70 to 100 nanometers in size. The microcrystals comprise about 30% of the protein in the fibers arranged as a beta sheets. They contain trace amounts of calcium ions, are about in the 70 to 100 nanometer crystal size of the crystalline. Professor Lynn Jelski of the Cornell University established that polyala repeats are involved in microcrystal structures. She fed spiders with C13 labeled alanine and studied the spider chains by NMR studies. The process of spinning converts the silk proteins from soluble alpha helical proteins to fibrous form containing beta sheet structures. Similar to soluble amyloid and prion proteins to insoluble fibrous form. The crystal part gives strength, and mesh-like part, which is less ordered structure, gives a flexibility. Understanding and manipulating the structural details of these regions might result in fascinating new materials. So students, let's summarize what we have learned in this module. Fibrous proteins are structural proteins. These proteins could be made up of helical structures or extended structures like alpha-keratin, which are coil-coil protein, consist of alpha helices. These are structural proteins, building blocks of hair, nails. The coil-coil structures are cross-linked with disulfide bridges, and the higher the cross-linking, the more rigid the protein is, the fibrous are. Collagen is made up of triple helix structures where three proteins form left-handed helices are twisted together into right-handed triple helix superhelix structure. A cooperative quaternary structure is established by many hydrogen bonds. Almost every third amino acid is glycine, which comes in the center of the triple helix. Collagen is a building block of skin, bone, and junctions. Any mutation in collagen genes cause several diseases. There are different post-translation modification and cross-links in collagen fibers which gives strength to these fibers. Silk fibrins are mostly beta sheet structures. The beta sheet structures form a microcrystalline structures also contains of calcium atoms, mostly built by poly-ala sequences. Other regions form a less ordered mesh-like structures, where these crystals are embedded. The crystal part gives strength and mesh-like structure gives the flexibility. So here we have learned about three types of fibrous proteins of three different types of structures. One, alpha helical containing fibers, the collagen extended helical structures, a third structures, the fibrous structures of the containing beta sheets. Thank you.